Hi everyone, this is Ms. Ramani again, and for this lesson we will continue to learn about cell transport by focusing on active transport. So in contrast to passive transport, which does not use cellular energy as it moves molecules down a concentration gradient, active transport uses energy from the cell to move molecules against their concentration gradient. In other words, from an area where there's a low concentration of that substance to an area where there's a higher concentration of that same substance. A concentration gradient is a chemical driving force that moves particles from an area of higher to lower concentration. So when a cell wants to move particles in the opposite direction, against the concentration gradient, it needs to provide the energy to counteract this force, which it does in the form of a molecule called adenosine triphosphate, or ATP. So why would cells need to do this? When particles tend to move naturally down their concentration without the need for cellular energy, why spend energy moving things in the opposite direction? Well, there are many instances where this type of movement is needed. Let me give you a few examples. All plants need water and minerals to survive. This is why plants have such a vast network of roots that go so deep into the soil. Some of the outermost cells that make up these roots are known as root hairs, and they have long protrusions that stick out into the soil, which is important for the absorption of water and nutrients. However, some minerals like magnesium, which is needed to produce chlorophyll, are found at lower concentrations in the soil than they are inside the cell. This means that the cell cannot absorb them by diffusion. Instead, it has to use energy to actively transport the minerals into the cell. Since this energy comes in the form of ATP molecules, and since ATP is produced in the mitochondria, we tend to find lots of mitochondria in root cells. Another important site of active transport is in the intestines. Usually after a meal, there will be a higher concentration of nutrients in the intestine than in the bloodstream. When this is the case, the nutrients and water can be absorbed into the blood passively by diffusion, by facilitated diffusion, and by osmosis down their concentration gradient. This involves the nutrients diffusing from the interior of the intestine, called the lumen, through the intestinal cells and into the blood vessels. However, as some of the nutrients are absorbed, the concentration gradient decreases and diffusion can no longer work. At this point, the cells lining the intestines have to actively transport the nutrients from the lumen across their membrane. And once they're in the cell, they can diffuse into the blood. And this process allows us to absorb almost all of the glucose and other nutrients that we eat. Also, notice that the intestinal cells are adapted for this amount of transport by having lots of tiny protrusions on their surface called microvilli, which provide a large surface area. These cells also have lots of mitochondria to provide the large amount of energy required for active transport. In contrast to absorbing nutrients into our blood at the small intestine, active transport serves an important role in our kidneys when it comes to removing waste from our blood and reabsorbing the nutrients that we need to survive. When urine is produced, we want that urine to contain our waste products and not our nutrients. So the kidneys use both passive and active transport to ensure just the right stuff is removed out of our blood and placed into the fluid that will become our urine. Another very important use of active transport is the transmission of nerve impulses. A nerve impulse is an electrical current that travels through nerve cells called neurons. In order for the electrical signals to work, the neuron has to first establish an electrical voltage by accumulating or concentrating different ions on the inside and on the outside of the cell using an active transport protein called the sodium-potassium pump. The sodium-potassium pump is actually a very important transport protein that is involved in a form of active transport called primary active transport. In this type of active transport, protein pumps act as enzymes, catalyzing the conversion of ATP into ADP, which releases the energy needed to transport ions against their concentration gradient. The sodium-potassium pump works like this. When the inside of the protein is oriented towards the interior of the cell, 3 sodium ions bind to it, and an ATP molecule provides energy to change the shape of the protein. That change in shape moves the sodium ions towards the outside of the cell. One phosphate group from the ATP remains bound to the protein. The sodium ions are then released on the other side of the membrane, and that changes the shape of the protein so that now increases the carrier's affinity to potassium ions. So now 2 potassium ions bind to the protein, which again now causes a change in the shape of the protein, which moves the potassium ions towards the inside of the cell and releases the phosphate group. This release allows the protein pump to revert back to its original shape, which has less affinity for the potassium ions, which are then released into the cell. And in that original shape, the protein pump now has a high affinity for sodium ions. And then when these ions bind once more, they initiate another cycle. The important thing to note about this type of transport is that both the sodium and the potassium ions are moving from areas of low concentration to areas of high concentration. Each ion is moving against their concentration gradient, which is why ATP was needed to provide the energy and change the shape of the protein pump in order to drive this transport. At this point, there are more sodium ions outside of the cell than inside, and more potassium ions inside than out. But for every 3 ions of sodium that move out, 2 ions of potassium have moved in. So this difference in the number of ions being moved in and out of the cell, combined with the fact that the interior of the cell has negatively charged proteins, results in the interior of the cell being more negative relative to the exterior of the cell. This difference in charge is actually very important because it maintains what is called an electrochemical gradient in living cells. Okay, so far we've explored transport through the membrane. Now let's talk about what happens when substances don't necessarily move through the membrane, but instead get wrapped up by the membrane. So let's explore bulk transport, or transport of substances in and out of the cell using vesicles. This type of movement can be divided up into 2 main types. One that moves substances out of the cell by fusing vesicles formed inside the cell with the cell membrane, and another that moves substances into the cell by forming vesicles at the cell membrane. So let's start by talking about endocytosis, the process by which substances are moved into the cell using vesicles. And there are 3 types of endocytosis. One is called pinocytosis. During pinocytosis, the cell brings liquids with dissolved particles into the cell by forming vesicles. And for that reason, pinocytosis is sometimes referred to as cell drinking. Another type of endocytosis is called phagocytosis. During phagocytosis or cell eating, the cell brings in larger objects like food or bacteria into the cell. For example, when microorganisms invade the human body, a type of white blood cell called a neutrophil will remove invaders through this process. The plasma membrane will surround and engulf the microorganism, which is then destroyed by the neutrophil by fusing the vesicle containing the bacteria or other pathogen with a lysosome. In many single-celled organisms called protists, phagocytosis is used as a means of feeding, providing part or all of their nourishment. Amoebas, for example, are a type of protist that uses phagocytosis in order to eat other smaller protists or bacteria. Unlike pinocytosis and phagocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to a specific molecule that are found outside the cell. Once these molecules are bound to the receptors, the membrane forms a vesicle that contains the receptors and their cargo and brings both into the cell. And then in contrast to endocytosis, we have exocytosis. Exocytosis is the process by which substances are moved out of the cell using vesicles. The vesicles arrive at the plasma membrane filled with substances that were produced by the cell. The vesicle, which is made up of the same phospholipid bilayer as the cell, then fuses with the plasma membrane. Exocytosis happens a lot in cells that are part of the excretory tissues or organs. For example, mucus-producing tissues, or cells in the pancreas that produce hormones and enzymes that need to move out of the cell. Exocytosis also occurs at the end of nerve cells called neurons in order to release special proteins called neurotransmitters that nerve cells use to communicate with each other. And so that's it for cell transport. And to summarize, movement across membranes can happen via passive or active transport. Passive transport, such as osmosis, simple diffusion, or facilitated diffusion, does not require extra energy from the cell. And active transport does require energy from the cell, both during primary and secondary transport, and in the movement and formation of vesicles during endocytosis and exocytosis. And that's it for this topic. Talk to you soon.